It's a full-on device, but it's a bacteria.
Yeah. A lot of failures, but with failure comes success at the end.
This is yogurt. Most people know what it is, it's delicious, and the good news is we don't need any prior training in biology to eat it or even to make it. In fact, there's a good chance that the first yogurt happened by accident because the bacteria that makes yogurt is basically everywhere. However, to actually decide to make yogurt with specific strains of bacteria, in a way, one could argue that that is one simple, very simple form of synthetic biology. Synthetic biology is a field of science where the researcher tries to re-engineer life. It adapts concepts from molecular genetics and physics and chemistry and engineering and even computer science in order to make things that are sometimes practical and useful and sometimes just plain beautiful. Today we're at BMC, or Uppsala University's Biomedical Center, and we're going to meet the iGEM team for Uppsala University. So iGEM stands for International Genetically Engineered Machine, and in this competition students from different disciplines and from different levels compete in synthetic biology.
This year we're developing a cellular biosensor. The idea is that we will have it adaptable for any kind of analyte that people would be interested in detecting. So whether it's a small molecule that is a pollutant in the environment, or it is a pathogen. We call it system nanoflex because the part that is modular and that is flexible and that we need to exchange to be able to diagnose or detect anything is a nanobody.
This year's iGEM team in Uppsala is trying to create a biosensor to detect different types of pathogens. They're using different approaches that rely on nanobodies, so I think it's important to know what a nanobody is. But before that, I think the beautiful thing about synthetic biology that I want to say here is that in their project they have this modular approach to the biosensor that they're building.
The signal detection, signal amplification, and the final signal.
I'm Ruben. I study applied technology. Here we are working with one of the detection proteins. We have tuned them so that they respond to presence of caffeine, but ideally we can detect anything. But caffeine is just a good molecule to do. We have these two tubes. They contain the same cells, but on these ones we did not add any caffeine, and in these ones we add a relatively high concentration of caffeine. And we see that there is a difference in expression of the red signal. So we are detecting a particular amount of caffeine.
Of course, one of the things that they had to explain to me is what a nanobody actually is. So we've probably all heard what an antibody is because of the coronavirus and how antibodies bind to pathogens and that helps the immune system detect them. So you could think of a nanobody as a much smaller version of an antibody. Other than just the size, nanobodies are usually more stable to heat, much more stable to heat, and generally much more stable at a higher pH.
Since it's much simpler, it can be expressed in a completely different organism, such as bacteria.
There is speaker? Yeah. Well, then you have me.
My name is Samuel. I'm going to start the third year of the same program we're studying. What's it called in English? Molecular biotechnology engineering.
Yes.
A mouthful. We're talking about different proteins in the lab, and we're discussing tuberculosis, for example. Currently, if you're able to get like tuberculosis nanobodies and like how that would work in the lab and stuff like that.
So I'm Nicole, and I'm from Germany. And I studied bioscience in Heidelberg before and came here for my master's.
So I have some plates where we transformed bacteria yesterday. So we have been working hard.
Yeah, I'd say that.
I'm Pedro. I came from Portugal, and I decided to come to Sweden to study immunology and microbiology. I like working in the lab mainly because we first do the theoretical part and then we get to see if it works. That's why the lab is so exciting, because we get to see results of the experiments that we are doing based on what we hope that will work.
It's really hard to plan those experiments. And many of the things that you're saying now are in theory, not yet proven, and will probably to some parts not be possible to prove before the deadline. So part of this is to be optimistic and see, you know, we could do this, because if you don't explore and dream, right, nothing's going to happen.
So tuberculosis is not always active, it could be latent, and it's really hard to detect it in the latent phase. What decides what we can or cannot detect is what kind of proteins or other biomarkers the bacteria leaves in human body.
The fact that you can see the pink color is actually important, because the fact that the output is visible to the naked eye means that this system could potentially be used at point of care, which means that you don't really need PCR machines or X-rays or people that actually know how to use these technologies in order to detect certain pathogens such as tuberculosis. I'm not even filming right now.
I have to put on the laptop.
If you have any thoughts or comments, please leave them down below. Thank you for being here and have a good day. Bye.